Hello, my name is Mahin Tachur, and today I'm going to give a presentation on expression and functional analysis of MELK in mammalian cancer cell lines. What is MELK? MELK is a maternal embryonic leucine zipper kinase, and it is a serine kinase that plays a crucial role in cell cycle progression, apoptosis, and cancer development. It is highly expressed in various malignancies, including breast cancer, glioblastoma, and colorectal cancer, where it is associated with increased proliferation and resistance to apoptosis. Given its potential as an oncogenic driver, MELK is a promising therapeutic drug. So the aim of this project is to clone and express MELK in mammalian cancer cell lines and assess its impact on cell proliferation and survival. By overexpressing MELK, I aim to determine its role in cancer progression and explore potential downstream signaling pathways that it may regulate. To achieve this, first, we will clone the cDNA sequence of MELK into a mammalian expression vector that is going to be pcDNA3 and transfect into a selected cancer cell lines. I'm using HeLa here. Functional analysis will be conducted through, again, Western blotting and cell visibility assays to evaluate MELK and its biological effects on the transfected cells. Uh, so, um, so next we have for the successful expression of MELK in mammalian cells, we have chosen pcDNA 3.1 plus, which is a high efficiency expression vector containing a CMV promoter, which drives strong and constitutive transcription in mammalian cells. This vector also includes a neomycin resistance gene, which allows stable cell line selection after transfection using G418 antibiotic selection. And then we also have the SV40 poly A signal that is like it ensures proper mRNA stability and translation. The cDNA that I obtained from NIH, this is the forward primer, reverse primered it kind of contains almost 22 nucleotides. This is the former primer where I have added the restriction site and the Kozak sequence that's there to enhance the translation efficiency. For the reverse primer, we have the sole site here as a restriction site, and then we have the stop codon. The cloning strategy is the amplification of MELK cDNA using polymerase chain reaction. After that, once the PCR amplified MELK fragment is obtained, both the pcDNA3 vector and the MELK insert will be digested using the selected restriction enzymes. The digested products will then be ligated using T4DNA ligase and ensuring that MELK is inserted in the correct orientation for efficient expression. After that, it will be transformed into competent E. coli cells for plasmid propagation. Bacterial transformation will be performed using the heat shock method, where the ligation mixture is incubated with competent DH5 alpha E. coli cells, followed by 42 Celsius, followed by the heat shock at 42 degrees Celsius for 30 seconds. Then we're going to transform the bacterial cells on agar plates containing the neomycin as the pcDNA 3.1 plasmid contains and resistance genes for the resistant feed to the bacterial selection. To confirm that the MELK gene has been correctly inserted into the vector, plasmid mini-prep will be performed to extract plasmid DNA from the selected bacterial colonies. The extracted plasmid will be then analyzed by restriction digestion and Sanger sequencing. We are using bidirectional tube sequencing here. Once the MELK pcDNA construct is verified, it will be transfected into HeLa cells using liposuction. After transaction, cells will be subjected to G418 selection to generate stable MELK expression cell lines. To confirm successful MELK expression in mammalian cells, we will perform Western blot analysis using an anti-MELK antibody to detect the expressed protein. Additionally, we can also use RT-qPCR. To assess the impact of MELK expression on cancer cells behavior, functional assays will be conducted. Here I'm using cell visibility assay or MTT assay that is going to measure the metabolic activity of cells to determine whether MELK overexpression promotes or inhibits proliferation. That's it. Thank you. This is from my side. References that I have used in this presentation.